Good morning, everyone. I'm James Poulter, and I'm from the University of Leeds. There's a little kind of brackets around the doctor, because I'm not actually a medical doctor, or as my wife says, I'm not a proper doctor. I'm a scientist. I do not come to me for medical information or advice. I'll point you in the direction of someone who actually knows what they're talking about. So this is day 2 of the community weekend, but actually on Friday, me and about another 60 or 70 research professionals were here doing a research study day. So this is day 3 for us, but still, it's all worth it, and we love doing it. So every year, Karen asks me to give a summary of what happened on the science day, and it's probably the most daunting talk I give, because I can't prepare for it really until the day before I actually give the talk. So there's nothing I can do in advance. And I'm going to be presenting work from other people. A lot of these, I'm not really an expert in that field, so please forgive me and bear with me as I go through the slides. Now, Karen said a lot of people are new here this year, and I guess it's really a good thing to really delineate what is research. It sounds like a silly question, it sounds like a simple question, but it really is a key thing of kind of what I want to get across today. Now, there's things we know, and unfortunately, there are things that we don't know. And actually, that's as big as I could get that circle. Actually, that circle's probably a lot bigger. And it kind of comes from that very famous phrase from, I think it was Donald Rumsfeld, who said there are known knowns, there are known unknowns, and there are unknown unknowns. And that's kind of, the known knowns are actually quite a small fraction of what we know. So, a lot of the things we've been told this weekend and you've heard are all, these are what leukodystrophies are, and these are all the different types, and this is what the MRIs look like. And all the things we know. But there's also a lot of things that we don't know. What's the future look like? And we'd love to know that information. We just don't know that information yet. So the whole point of research really is to take that very large unknown and make the large unknowns into, a smaller unknowns and things we know much bigger. So that's the whole point of research, really. We want to make that circle on the left much bigger and the circle on the right much smaller. So how do we do that? How do we do that quickly? Because we don't want this to take hundreds and hundreds of years. We want this to happen ideally in the next year. So we do this once next year, and then we're done, really, for research. Obvious one, money. And we heard from Sarah yesterday how difficult things are in the charity sector in terms of raising money, and it's much the same in every sector. So in the science and academia research sector, it's very challenging at the moment trying to get money to do this stuff. Really, we need to share knowledge, okay? We're not all experts in everything, okay? We need to get all a kind of different people together, find out what they're doing, what works for them, what doesn't work for them, share that information so that we're not all repeating ourselves and all doing the same thing and getting nowhere. We need to identify the gaps in the knowledge. What don't we know? How do we find out that information? How do we get to that point where we know what's going on? And much like you see in the clinic, we need to bring multidisciplinary teams of scientists together, okay? And that includes the clinical teams as well, okay? But it's also about, I'm a geneticist. So I can tell you lots about genes and what they do, but I'm not a chemist. I can't make a drug. I don't know how to make a drug work better. So we need to bring all of those teams together to really try and accelerate the research that we're doing. And so that's kind of why a few years ago, in the COVID-19 pandemic, when we didn't really have much else to do, we made this network of leukodystrophy researchers in the UK, because we were all kind of working in silos, didn't really know what each other was doing. And so, we thought if we actually try and bring people together, it would make things a lot easier for us to understand and also accelerate the research that's going on. If I find something interesting from a genetics perspective, I could share that with Noemi, who's now in York, and you might remember the fish last year. If you were here last year, we had some zebrafish here. She could look at what that gene does in a fish, and Sean Sweeney, he could look at what it does in a fly, and we learn different things from looking at all these different models. And obviously we kind of bring in Lydia and all of the clinical teams as well, because it was only yesterday that I found out that the white matter is actually black on an MRI. That's the first time I never knew that, and I've worked with Lydia for a good 10 years now. So, there's lots of things we don't know. I think that was an unknown unknown. So the whole point of Luca Labs is to improve the lives of individuals affected by and impacted by leukodystrophies. And so, much like the things that we need to do is what we're trying to do. We're trying to unite researchers and clinicians with a common interest in inherited white matter disorders. We want to promote collaborative projects, not just around the UK, but around the world. You know, Europe, it's a little bit trickier now to work with Europe and internationally now post-Brexit, but, you know, we can still keep trying. We want to understand why and how leukodystrophies occur, how they develop, the future, develop therapies. Obviously, that's the ultimate aim of what everyone wants out of this. To try and attract more funding, and in the last year alone, we've probably put in over £10 million worth of funding applications from the network alone. Alex TLC have been part of a lot of them as well, so we thank Alex TLC for that. And what's really important in terms of the future of research is to develop the next generation of researchers, so to get young researchers interested in leukodystrophies. And we've had some really exciting examples of them at the Science Day this week. So you might remember the windows from last year, if you were here in Birmingham last year. You know, scientists are a little bit like vampires. It was a bit bright for us. And we like blood as well, blood samples, so it's, you know, it's a very close link. And so this was the first Science Day we held last year. The year before, I think we did a 2-hour session on clinical trials, but last year we did a full day on the Friday, and it worked really well. People who attended said they liked it, they enjoyed it, and so they made me organise it again this year. So this is this year, fewer windows this year, but the same kind of room full of people who were all interested in leukodystrophies from all around the UK. And we had from Europe, we had some American researchers and clinicians joining us too. So, you know, this is about bringing the UK research field together, but it's also about connecting out worldwide as well. And obviously, we're very inclusive and welcome all of those researchers who are interested. So what does a Science Day look like? Or we should really call it Research Day because there's clinical things in there as well. It's not just science. So we do split it up into 3 sessions. We have a clinical session, and that's kind of about, you know, how we can improve the clinical care on a daily basis for patients, potential different therapies and different regimes, different things that have been tried, what worked, what didn't work. You know, because if someone's trying something down in London, you know, the teams in Leeds need to know the same things, what works, what didn't work, because it's a lot of these things, you know, all leukodystrophies are rare. So, you know, the size of the trials that are being done are very small, as you know. So, you know, that information sharing is really key. So kind of facilitating that and getting people to come and present their findings is really crucial. Think about how do we assess disease progression with and without treatment. We've heard a lot about how variable leukodystrophies are. You know, the same patients, the same family members with the same mutations, same disease, can present very differently. Okay, so how do we predict the future, the prognosis of that? But also, if we, you know, try a drug, put them onto a clinical trial, how do we know if it's working or not? You know, if somebody is very stable on a clinical trial drug, would they have been stable if we hadn't given them that drug? You know, if they're stable but they're having some side effects, are we actually making them worse? You know, so how do we measure the impact of a successful drug? What does success look for in drug? And so there's a lot of discussion about this, not just in leukodystrophies, but in rare disease in general, because, you know, if we're thinking about cancer, there are unfortunately a lot more patients with cancer, you know, much bigger numbers to see that kind of prognosis and predict that, whereas with rare disease, we're often looking at, you know, less than 10 individuals. We also had posters. You might have seen some of the posters that have been left up outside. You know, not everyone had the opportunity to talk, which is fantastic in a way. We've got more people who want to come here and present and talk about their research than we could actually fit into one day. We had a keynote speaker who was actually really interested in myelination, but from the perspective of multiple sclerosis. So she had not really come across leukodystrophies, and yet her field of research was so relevant to what we were doing. So, you know, it's really important to kind of bring those kind of leading experts from around the UK here and say, look, this is what leukodystrophies are. Your research is really relevant to what we're doing. We want to kind of work together, and she was fantastic this year. We had a parent and family presentation, so many of you will know Gil. I don't know if he's in the room, but, you know, he gave a really interesting insight into parent advocacy for research, and I think he gave the same presentation yesterday as well as part of the weekend. And we have lots of discussion sessions. So it's a lot to fit into one day, but it's very much kind of inspiring. And obviously there's lots of coffee and cake. I think you've all seen how much coffee has been out here this weekend. It helps a lot. And so we don't think there's actually anything like this for leukodystrophies anywhere in the world. This is, to our knowledge, the only place where all of those different kind of expertise come together. And really, it's Alex TLC that helped us bring all this together. I mean, Suzanne, sorry to put you on the spot, but she's organised the 2 days of the community weekend. She helps us organise the science day as well, and answering all of the emails and organising everything. So I really do want to say a massive, massive thank you to everyone at Alex TLC for helping us organise the science day as well as the whole weekend. So we really couldn't do it without them. So I'm not going to go through all of what we spoke about, but I just wanted to give an example of what we were doing. So, in terms of the clinical sessions, we are looking at, taking small, well-defined groups of patients, doing what I mentioned earlier, following them, seeing how they present, how they progress, trying to understand and kind of get to that point where we can start to predict better prognosis and give more accurate kind of more accurate prognosis. Again, as I mentioned, we've heard a lot about Aicardi-Goutieres syndrome and baricitinib, and so we heard some of the early clinical kind of trials and outcomes from those, as well as kind of a result of about 6, a small cohort of patients who had undergone stem cell transplantation for Krabbe disease, and kind of what are the outcomes of those, are at least the early outcomes. And as I mentioned earlier, Amy Donald from Manchester was really talking to us about how do we design good studies to understand whether a treatment has worked or not. And this really is a huge limitation. We were talking about this 3 years ago at the Science Day, and we're still talking about it now because there's no easy answer to this. And it's equivalent of having a 400-meter race. So once around a racetrack, we've got a finish line, but if everyone starts at a different point around the track, and everyone has a different speed, how do you predict who's going to win and who's going to get to the finish line? And if someone doesn't get to the finish line, is it because they're much further away, or is it because they were slower? And the problem we have for clinical trials is there's one defined endpoint, and if it doesn't meet that endpoint, it doesn't pass. It's seen that it hasn't met that it hasn't met that primary outcome. So we need to make sure that we're designing those clinical trials well, so that the outcomes are meaningful, the endpoint is something meaningful to the patients, but it's actually something that's achievable. And if there is an improvement and it does have a beneficial impact, we need to be able to measure that and make sure that we can get that information across, so that when we go to the FDA or the drug trial companies, they want hard facts, and just saying, well, it kind of had an improvement in some and not in others is not enough to get it approved for clinical use. So it's a really important field that we need to really keep working on. We had a science session, we had some posters outside, we had 3 talks, and these are really kind of exciting up-and-coming researchers in the field of leukodystrophies. And they were talking about how cells work, how the different molecules work within cells and how they measure these. How do we make better tests for adrenal leukodystrophy? So Triana is looking at a 5-minute test which will tell you whether your kind of ABCD protein is working or not. Rather than waiting a year for a genetic test to come back, waiting a few weeks for an enzyme test, can we do it in 5 minutes instead in a quick blood test that you could have in clinic? That's the kind of point we want to be, is while you're still there in the clinic, can you actually get a result back? You shouldn't really want to be waiting years to get an answer. And there was also mention yesterday about gene editing, and some early work going on here about doing that. Can we use gene editing to correct genetic mutations to basically correct the mutation in utero, even kind of in the womb before a child's even born. So these are very early studies. These are preclinical, nothing kind of at clinical stage, but we need to start somewhere. We need to start understanding how these things work and what's possible and what's not possible. And then the final session is our clinical trials update. And again, you've probably heard of many of these companies. Minox presented yesterday. And so I'm just going to give you a very few updates from these clinical trials, because I think there's obviously what's relevant to you and what you're interested in, really. So BridgeBio, this is the Canaspire trial. This is led by Professor Florian Eichler. And this is a gene replacement therapy, so they put a functional copy of the ASPA gene in by a blood transfusion. And they've been doing some early clinical trials, which have all been really promising. So they've been testing how safe this is, how tolerable it is, and kind of how the drug works in the body. And so that's ongoing, and they're very excited about the results from that. Orchard, again, probably all have heard of this pretty much for MLD. So this is where they take a blood sample and purify the stem cells in those blood. They put a healthy copy of the gene back into those cells, and then once they're happy that those cells are happy, they will put that back into the body. And this is the steps that they go through, and they've done this for a few different treatments now. And what's really exciting about this is the method is quite generic. It's well defined, it's well tested. So there's the possibility of putting different genes into those cells depending on which gene has a mutation in it. It's not specific to one particular gene. And again, the results from those early trials are looking really promising as well. They're all fully enrolled now, unfortunately, but they are doing the long-term follow-up, and they're following them up 10, 15, 20 years. And I think it was Eric yesterday saying, what we really want to make sure is that there's no long-term disadvantage of having these therapies. There might be a short-term benefit. We want to make sure there's a long-term benefit too. We also heard about Ionis. So Ionis have got an Alexander disease trial ongoing at the moment, but they're also moving into a few other leukodystrophies as well, which is encouraging. And they're looking at starting one early next year, hopefully. And this is for anyone with a PLP1 duplication. So this is for PMD, which is one leukodystrophy that I can't pronounce. I'm not even going to try. If you want me to try afterwards, I'm very happy to try. But hopefully that's setting up, and we'll get going next year in Leeds. And again, you've probably heard Aaron's talk yesterday if you're interested in leriglitazone for ALD. And although there's a phase 3 clinical trial ongoing in America at the moment for this, they have actually submitted the evidence to the European drug approval in Europe. So they're hoping by this time next year they will have an answer from them as to whether this has been approved for use in Europe, and then it'll be a few months after that in the UK as well. So all really promising. We've got clinical trials coming from beginning and some coming to an end, all of those with promising results. So the counter is still going. I've got 2 minutes left, so I'll try and finish in 2 minutes. I think really what I wanted to get across today is that the research in leukodystrophies in the UK really is going from strength to strength. In the last 5 or 6 years, we've gone from being a few siloed labs around the UK to a network, and a network that people want to be a part of and want to contribute to, and I think that's really exciting. There's still a lot that we don't know, but we want to find that out. Okay? So we are very determined, stubborn individuals. We don't get out much, but there's good reason for that, and that's because we don't like not knowing things. We don't like saying we don't know, just as much as I'm sure the clinical teams don't like saying we don't know. So we really do want to find out. And I think, again, we've heard this all weekend, leukodystrophies are clinically quite challenging. They're very variable and they're very difficult to predict, and we don't like that. And scientifically, you can probably see where this is going now. Leukodystrophies are challenging, they're variable, and they're difficult to predict. And from a clinical trials perspective, they're challenging, they're variable, and difficult to predict. So, as a group of conditions, they are very challenging to work with, but that doesn't mean that we shouldn't be working on them. It just means that we need to work harder and bring in more funding to do it. So going forward, Phil and Kirsty would say location, 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 and this has been very nice in Wembley today. It's the closest I've actually been to Wembley Stadium. I actually went out and took a picture of it just to prove I was here. I don't usually actually get out of the hotels in conference weekends, but it's nice to do that. But the collaboration is a wide collaboration. It's clinicians, it's the nurses, it's the specialists, it's the scientists, it's the pharma, it's Alex TLC, it's you guys, it's the families, You are the driving force and the inspiration for what we do. If it wasn't for you guys, we wouldn't really be here where we are now. So we really wanted to say thank you to the patients and their families. We understand that clinical trials and being part of research isn't easy. You know, at times it's scary, it's very inconvenient if you have to travel to a different country or even just halfway across the UK, it's not easy. And I'm sure for, you know, being a carer or as with a family member, it's heartbreaking as well. So, you know, we hope we can repay you back for what you're giving to us, and we hope that we can kind of improve the lives of all of you if we can. So thank you. Happy to take any questions. This is flashing at me now, so I will stop there.
Thank you, James. It's always great to hear your summary, and thank you to all of you, you know, especially you and Dr. Green for all the work, and Naomi. I know she can't be here with us today, but you know, there's a lot of work goes into organising the Science Day. So you know, huge thank you to you guys as well. Does anyone have any questions for James? Yes?
Can we put the microphone on for the question?
Is it working? Okay.
Yeah.
Hiya, thanks. Thanks, it was a great presentation. I always find definitely with ALD specifically, sometimes it feels the UK isn't as involved in the research. So it's really nice to see things like Leuco Labs. I just wondered if you see UK research growing at all or expanding, or specifically with Leuco Labs, do you think that's something that will keep expanding over the years?
Yeah, absolutely. I mean, that's one of the main things that we want to do is to really make the UK a centre for leukodystrophy research. You know, there are, as you say, America is very big in that field, and some of that is purely the way that their funding is given to them.
Yeah.
And the same in Amsterdam. You know, there's a big group of leukodystrophy research groups there, and they all do something similar. You know, they have a big network and they all interact with each other. So what we really want to do is replicate that and make it even better. So you know, we're not going anywhere. I've got another 30 years to go before they'll let me retire. So I'm hoping at least for the next 30 years, but by bringing along the next generation of researchers as well and getting them interested in leukodystrophies, you know, they're going to take what we learn and make it even better. So yeah, we're not going anywhere anytime soon.
Could I cheekily ask on top of that, what are the main barriers for UK research? I know that obviously funding works very differently in places like America, but I know, is it universities that primarily get involved in research in the UK?
Yeah, so well, there's two main areas. There's the NHS and what's called NIHR, so that's the NHS research arm of what they do. And yeah, research universities, so academic universities. So most of the funding comes from charities or the government. And obviously, as we heard yesterday, you know, the government and the charities are both struggling for money themselves. And so, you know, for us then, we're all applying to the same smaller pots of money to try and get that funding to come in. So, you know, there are other routes, but those are the main two routes of funding. We try and work with the industry, so the pharma companies. Yeah. They often have a different mode of attracting finance. And sometimes we can tap into that funding as well, if it's something related to the drug that they're developing. But yeah, I mean, money and expertise, and so that's what we're really trying to build upon. You know, if we had infinite money and infinite people working on this, then things would move a lot quicker.
Yeah.
But there's still time.
All right, fabulous. Thanks so much.
Thank you.